Good morning or good afternoon, everybody. Welcome to today's webinar of the Critical Path Institute in collaboration with the PKD Foundation. Today we are talking about the TRxA Bridge Awards, again in collaboration with the nonprofit PKD Foundation. And the Bridge Awards, that stands for Bridging Research and Innovation in Drug Development Grants. So I'm sure you all are eager to find out more about this grant opportunity for you to apply to. So let's go ahead and dive on in. So first of all, quick background on what TRxA is. So TRxA stands for Translational Therapeutics Accelerator. We are a global drug discovery and development program, and we support academic scientists to advance new cutting-edge therapeutics from the lab to the clinic. So a few things of note. Global is underlined and bolded. We can fund projects anywhere in the world. Then academic scientists are a target audience. So when you are in a startup or biotech, you're unfortunately not eligible, but nonprofits and again, people in academia are. And then we are focusing on therapeutics in particular. So no devices or anything like that, but therapeutics is really where our sweet spot is. So what is new for this year is our PKD dedicated foundation track. So this is a partnership with the PKD Foundation, really designed to accelerate high quality translational therapeutics for PKD. So with this, I would like to invite Chris to come on camera and talk a little bit more about why the PKD Foundation decided to partner with the CPath and Critical Path and TRxA. And go ahead, Chris, and maybe share a little bit more about why PKD Foundation wanted to do this.
Great. Thanks so much, Maaike. So my name is Chris Chen. I'm the Vice President for Research Programs at the PKD Foundation. I just wanted to take this opportunity to express the Foundation's enthusiasm for this partnership with TRxA. We can go to the next slide. I wanted to start with some background. For those who aren't aware, there's only one approved therapy for autosomal dominant polycystic kidney disease. It's called tolvaptan. And tolvaptan is a drug that slows the growth of cysts, and in doing so, it delays the onset of end-stage renal disease, which requires the need for either transplant or dialysis. So there's a lot of room for improvement in terms of more effective drugs that may prevent the progression towards end-stage renal disease, drugs that may stop cyst growth, or even drugs that could shrink cysts and reverse disease. There's also a lot of room for improvement in terms of drug tolerability and safety. So I think the Foundation's done a really good job at enabling proof of concept types of studies that have led to a lot of potential therapeutics that have performed better than tolvaptan in these very early studies. But the Foundation's grant program really hasn't been set up historically to enable research that supported those studies further in progressing towards clinical trials. So having met Maaike last year and learning about the TRxA grant opportunity is really just fantastic to build upon the partnership that we already have with the CPath Institute as well as the PKD Outcomes Consortium, and to help fill this gap in terms of funding. It's been great to learn that Maaike and her team have an established program that we could take advantage of, build our partnership on. We don't have to build a program ourselves, and it's not really just the funding program that we're looking forward to taking advantage of. There's the expertise and mentorship side of things, which is really critical, I think, to our academic PKD research community, many of whom really don't have that experience of moving drugs through the preclinical phase and getting them ready for clinical trials. I also want to mention that on the back end, Surin Fidelis at the PKD Outcomes Consortium and we both meet with a large number of industry partners, many of whom do not have an asset in PKD, but have a very strong interest in getting into this space. So just to say that I think the outcomes from these TRxA grants and these projects are going to lead to the kinds of assets that we already know that there's demand for in terms of investment. So really just filling all the gaps and some critical areas of unmet need. And again, just to reiterate that I'm super excited for this partnership and looking forward to the applications that we get this year. Thanks, Maaike.
Fantastic. Thank you, Chris. Yeah, we're excited to have a partner in the PKD Foundation as well. It broadens the types of projects that we can support together. And let's dive in and what we do and how we operate. So with this, I would like to invite the TRxA team as a whole to come on camera and do some brief introductions. So my name is Maaike Everts. I'm the Executive Director of TRxA. But then Mark, would you like to introduce yourself?
Yeah, my name is Mark Drew. I'm the Director of Drug Discovery and Development at TRxA. I've been here about 3 years, so excited to see the applications.
Fantastic. Michelle.
Hello, everyone. I'm so glad you could join us today. I am Michelle Morgan. I am Associate Director of TRxA, and I oversee the operational aspects of our program.
Fantastic. Kyla.
Hello, everyone. Thanks for being here today. My name's Kyla Edding. I'm Project Coordinator for TRxA, and I help Michelle with the operations side of things.
Great. And then last but not least, Samantha.
Hi, everyone. Thanks for joining today. I'm Samantha Wilkins, and I'm a project manager with TRxA, and I've been supporting the TRxA teams and their projects for about a year now.
Yep. So when anybody connects with us, one of us will be answering your call, your email, your LinkedIn message. So we all really are very well connected amongst each other. So if one of us doesn't know the answer to one of your questions, we'll ping somebody else from our team and get back to you as soon as possible. So with that, thank you, team. I'll go into the meat and potatoes of today's webinar and start with the drug development gap. So the reality is, as Chris kind of alluded to earlier as well, that promising academic research with the potential to really transform lives very often stalls before reaching the clinic. And there's a lot of reasons for that, but you really need coordinated funding, infrastructure, and also regulatory expertise to move forward through this valley of death. And that really is kind of an underfunded area typically. It's also an expertise gap for many people that are in academia. So this is where we hope to help you out and really shepherd your project to the finish line. One of those reasons why this is so hard is it's very complex in this translational science space. So on the left is a figure I do not expect you to be able to read, but I do have the reference on the right bottom corner. Chris Austin wrote this paper a couple of years ago in Clinical and Translational Science, and it really talks about the interplay of all the different disciplines and ecosystems that need to come together to get something through the valley of death. So most of us in academia probably live in this basic science research and target identification realm, which is depicted here on the upper left corner. There's also target pharmacology and biomarker development, still fairly comfortable for most of us, I would say. And then there's lead identification, like come up with a molecule that may interact with the target. Then you need to optimize it, select candidates, IND-enabling studies, scale up. There's things like formulation development, salt forms, manufacturing. There's a lot of different aspects to drug discovery and development that are typically not in the realm of an academic investigator. So with all of that, that interplay is so important that accelerators like TRxA can really help reduce risk and make sure all those pieces talk to each other. So one of the main things that accelerators can do is bring experts together early. So scientists like yourselves, clinicians like yourselves, but also industry partners can having maybe a toxicologist work together with you, somebody who understands manufacturing, somebody really understands the patient perspective. Everybody has to communicate early on to get your project set up for the best success possible. Now, you really need to focus on the right pharmacological target, really making sure that the science is aimed where it can truly impact patients. And accelerators like ours typically do that by doing a lot of diligence on the applications that come in. And together, we really work to strengthen your research plan. So we add this outside input into your plan as you propose it. Together, we refine experiments and really avoid dead ends. Then, of course, we need to provide rigorous testing. So you would probably evaluate your new therapeutics in your animal model of choice, maybe with a collaborator. What also very often is important is that you have a third party that's neutral, that replicates a key experiment that shows that your therapy works in an animal model. This, when you go to the next step and you want to get funding from, let's say, a venture capital group, or maybe a licensing agreement with an existing biotech or pharma, having an independent group having replicated your experiments is going to add a lot more confidence in your data package. And this is robust enough to really have a meaningful impact ultimately in patients. With that said, we really want to engage and stimulate engaging industry early. So you can align promising projects with what pharma and biotech would expect of a data package before they would write a check to you. And getting that feedback early, if you start to talk to a lot of different groups and they all ask the same question, well, that is, of course, a flag that you probably need to have an answer for that question before anybody would further invest in your project. And then finally, we really want to build your program with the regulator in mind. So really laying the groundwork for future approval from day one. So even though you may live in a preclinical world, maybe in vitro, maybe some in vivo experiments, talking to patients, talking to clinicians, thinking about how would you set up a clinical trial? What would the endpoints be? This is really critical because it may inform some decisions that you need to make on the front end and set up your project for success. So not all accelerators are the same. So some unique highlights from CPAT's Translational Therapeutics Accelerator are on this slide. So we have a global reach. Again, we can fund projects anywhere in the world, supporting academics to advance therapeutics from the lab to the clinic. So we are not a commercial accelerator program. We do not take any ownership in your IP, for example. We are a nonprofit and we're really patient focused. And what's really important too is that we're powered by CPATH. So CPATH stands for Critical Path Institute. It's a nonprofit that has existed for 2 decades. Actually, we just turned 21 in 2026. And we bring together scientists, regulators, and industry to help accelerate safe and effective treatments to patients. So we have a long track record of improving the ecosystem and bringing together a lot of different groups to come up with drug development tools that really speed up the process to get new therapies to patients. So a little bit more about CPATH. So we work in what we call public-private partnership models, and the PKD Foundation is part of such a partnership called the PKD Outcomes Consortium. Those kind of consortia really foster development of new evaluation tools to inform medical product development and regulatory decision making. So how we do that is we convene these consortia where a lot of stakeholders come together, industry, academia, patient groups, government, and everybody shares data and expertise. And what it allows us to do is build consensus around what is the critical obstacle in this particular therapeutic area that together we can overcome. We share the risk and the costs. We get the best science on the table and have the broadest experience. And what's really critical too is we have iterative participation of regulators around the world. So FDA, EMA, PMDA for Japan, and those individuals and those groups are at the table at these consortia. So you immediately have that regulatory mindset right there in the consortium to make sure that you're on the right track. And then ultimately, we like to get official regulatory endorsement of these tools so that they can be used in practice. So we've had quite a bit of impact over those last 20 years. So we have 40 formal regulatory accolades, lots of science and data contributors. We like to collaborate, lots of partnerships. We have 24 active programs and lots of publications. So it's really a nice ecosystem for us as a drug accelerator to live in because we can pull on the expertise from all the colleagues that we have. So how we then operate within this CPATH context is that these consortia that come up with these drug development tools, and their regulatory endorsements, they have a lot of expertise, especially on that typically clinical phase of development. And those consortia then provide for us access to subject matter expertise to help get your project through the valley of death and towards the clinic. So a little bit about our scope, because we cannot fund every research out there. So we have a couple of critical pieces that make you eligible for our program. So we talked about this a little bit already, but we're looking at therapeutic molecules. And these molecules need to be in kind of lead optimization IND enabling studies stage. It is important for us to have composition of matter, intellectual property, either in place or obtainable. You do not need to have IP in place in order to apply, but the goal needs to be development of strong IP like composition of matter. So that means that repurposing is out of scope for our grant program. There's a lot of kind of nuances and intricacies that come with repurposing and the business development, the business model around those. So for now, we are focusing on composition of matter type of projects, again, in place or obtainable. And that IP needs to still be within the institution. For us, it's important. We are a neutral convener, getting all those groups together within CPATH, so industry regulators, patient groups, and so on. So to avoid even perceived conflict of interest, it's important for us that we focus on academic or nonprofit groups that are doing drug discovery and development. And once it's in a for-profit entity, it could even be a startup, then unfortunately you are not eligible anymore. Quick highlight on who funds us. So we received a very generous grant from the Frederick Gardner Cottrell Foundation. This is established by the Research Corporation Technologies Group that's in Tucson, Arizona. It's a venture capital group. And their foundation has been established many moons ago in 1998. And they've, over the decades, provided financial support for both scientific research, but also educational programs at qualified nonprofits like CPATH. So we're very thankful to the Cottrell Foundation for setting this up. So then a little bit more about our model. Let's start on the left, how we begin. So we've launched disease-focused RFPs like we're doing now this year. And those RFPs are really aligned with funder priorities and investments. So for example, this year we have a separate dedicated track for polycystic kidney disease. And together we really want to build this global pipeline of academic discoveries. And the awards that we give out, and we'll talk a little bit more in detail later, but they range from US dollars, 250,000 to about $1 million. So you can convert that into your local currency. Again, we can fund projects anywhere in the world. So how we drive this then is that we, of course, have an expert review system. We do due diligence. And then the projects that we select, we set up milestone-based awards with go, no-go gates. And we work on those milestones together with you. We probably will propose something, you propose something. We kind of go back and forth a little bit and set up something that is reasonable and workable. Now, what's really important, what I think is kind of special about TRxA, is that we provide hands-on support. So we give you scientific, translational, and regulatory feedback. And that is done by monthly meetings. So when you get an award from us, we get together at least monthly and really talk about what's been done the last month, what should we do next. And these are not punitive meetings for you to tell us what you did last month, but it's really a collaborative team partnership so that you get kind of an extra set of eyes, brains, ears to help make your project as successful as possible. And we stimulate early industry feedback loops. So go talk to people that may want to ultimately license this from you. But what's important, us as a nonprofit, we don't want to own any IP. So the IP really stays with you with the ORD, so typically your university. And ultimately what the outcome is that we focus on drug development and there's a reduced emphasis on kind of traditional academic outcomes. So for us, it's most important that we get solid IP protected, maybe more so than, let's say, a publication. So there's a couple of consequences of that, and we have a whole guidance document that kind of talks about operating principles that spells some of those things out. But we really, again, want to focus on getting drugs to patients. Ultimately, in order to do that, you probably need more money than the money that we are able to provide to you. And so you need to talk to potential investors or licensees. So getting those data packages attractive to that next funder is really important to us. And this then increases the success for those follow-on funding licensing pathways and a higher likelihood of reaching the clinic. So a little bit more detail about the awards themselves. So for 2026, we have 4 groups of indications that are eligible. The first one is rare and orphan diseases. Then we have brain health, pediatric indications, and then of course the dedicated track for polycystic kidney disease. Do want to point out that for the rare and orphan diseases program, any rare and orphan diseases welcome. We do have a special co-funding opportunity with Project CASC. This is another nonprofit that we collaborate with. If there's a project in CASC that rises to the top regarding scientific quality and so on, Project CASC has committed to co-funding projects like this with us. And then same for brain health, in particular neurodegeneration. We have some co-funding opportunities with Cerebrum DAO. It's another nonprofit that has pre-committed to let's look together at neurodegenerative proposals that come in under the brain health banner. And if there's one that we both like, then we will co-fund those projects together. And again, those partnerships with the PKD Foundation and then these other groups really allow us to fund more projects. The modalities here on the right that we support are small molecules, protein-based therapeutics, and in vivo gene therapies. And we'll dive a little bit more into some details there because you probably have questions like what is eligible here. Now, when I talk about how much money you can apply for, we have 3 groups. So our stage 1 is really an early lead molecule to late lead series stage. So this is for up to $250,000. And the eligibility criteria differ a little bit depending on the modality, and we'll dive into those details in a little bit. Then you can apply for stage 2 proposal. This is for up to $500,000 and that is if you have a late lead series and you need to select a clinical candidate. Or in the case of a protein-based therapeutic, you have shown that it works in animal models, you've worked out some manufacturing issues, same for gene therapy. Again, we'll talk about those eligibility criteria in a little bit more detail. So it's a little bit more mature project than that early stage 1. And then we have a stage 3 proposal. We don't award too many of these, also based on our own funding constraints, but for select programs, if you have a candidate selected and you need to do IND enabling studies, if you've ever been in this field for even a little bit of time, you know this costs a lot of money. So you can apply for up to $1 million to help with those kind of expensive steps of the drug development process. So manufacturing comes into place. For small molecules, it could be route optimization to make sure you can scale it to, let's say, kilogram scale. For gene therapy or protein-based therapeutics, again, making sure that you can make enough of your vector or your antibody drug conjugates. You got your cell line set up. You need to maybe do GLP tox. All of those types of studies cost a lot of money, so that's why the amount of that award is quite a bit higher, but the criteria for these are fairly stringent. So then just a couple of examples of protein-based therapeutics that are eligible. Peptides or proteins are, of course, a part of this cohort. Then also antibodies. Regular antibody will be more than welcome, but also bivalent or maybe even cool new trivalent antibodies, antibody drug conjugates, protein drug conjugates, things that ultimately include an amino acid of some sort in your molecule, which would be eligible for protein-based therapeutics. For the in vivo gene therapy, there's also some examples on this slide. It could be a gene replacement approach. For example, what's done quite a bit is using the liver as a factory for a missing enzyme that the patient themselves is not making. In vivo gene editing, we do not allow ex vivo approaches. The reason for that is the manufacturing of that is quite complicated. We actually are big believer of the promise of gene editing, even using cell therapy and so on, but from our scope, we needed to narrow it down a little bit. Again, we do have ourselves some limitations there as well, how many projects we can support. So we are limiting this to in vivo gene editing. Gene silencing or oligonucleotides also would fall under this category, and we welcome projects using those modalities. So then let's dive into a little bit more detail about you might be interested in, like, well, how do I qualify for stage 1, 2, 3 of whatever kind of award? So a little bit detailed slides, but I think fairly critical to making sure you position your project for the right type of award. So for small molecules, a stage 1 proposal. So again, this would be for $250,000. What you need to have in place is a compound progression pathway. So what is the plan for your compound to be, to go through the different assays that you have, and what are the criteria where something meets those criteria or not? Do they go forward? Do they fall out? At this stage in the game, very often you will make tens, if not hundreds or thousands of molecules. So you need to have a very good filtering system to know which compound you allow to progress to the next assay. You also need to have a target product profile completed, and this actually is something that is in place, a requirement for any award that you apply for. So this is really a living document that talks about what is your patient population, what are the minimum requirements of a new drug. For example, how often do you need to take it? Is it once a day oral dosing? Is it, an injection every week? What are acceptable side effects? And this is really helpful. Here, it's really helpful to think about what is the current standard of care for your patient population of note. So again, talk to clinicians, talk to patients if you haven't done this yet, because it really helps you inform putting together a target product profile. So the project needs to be an early lead optimization. So you need to have done some work already to demonstrate an optimizable structure-activity relationship. This means that if you have done, let's say, high throughput screen and you have some hits, you're too early for us. You need to have done a little bit of chemistry. Now, it could be with commercially available analogs, but you need to show that there's a way to optimize your molecule without completely killing your activity. You need to have your in vitro assays in place, and they need to have appropriate throughput and turnaround time so that you can screen an adequate number of molecules. And then in vivo models need to be at minimum be available. There's no need to have animal data, but it is encouraged. And of course, IP needs to be either in place or obtainable. So then for stage 2, the same two items apply. You need to have a compound progression pathway. You need to have a target product profile. But here you're really looking at, I have a lead series. I've characterized in vitro pharmacology properties. I know my cell-based activity. I know in vitro ADME, so absorption, distribution, metabolism, and excretion, kind of those PK parameters. And you need to have some optimized candidates from ideally more than one series available. And you need to have really clearly defined what are the remaining optimization goals. There's probably something that needs to be fixed, whether that is selectivity, oral bioavailability, stability. There's all kinds of parameters that are just not ideal yet for a molecule that you want to ultimately treat patients with. So what is that you need to fix? And at this point in time, that lead series need to have shown at minimum target engagement in an in vivo model. And then the stage 3, so again, this is this highly coveted $1 million award. You need to have your target product profile in place. At this point in time, you really need to have a clinical candidate. It needs to have been selected with characterized ADME properties, and you need to have shown efficacy in an in vivo model. The clinical candidate also needs to have characterized at least some toxicology properties. You need to have a GMP scale-up plan and characterization plan in place, and you really need to have a non-clinical formulation that's been defined. So then moving to protein-based therapeutics. For a stage 1, $250,000 proposal, we're looking at having this candidate progression pathway again established with clear criteria, target product profile. The project really is in lead optimization. Again, in vitro assays need to be in place. In this particular case, you also need to have assessed the immunogenicity potential. With protein-based therapeutics, there's always this chance of humans rejecting proteins that are not natural to the body. So you need to have a de-risking strategy identified there as well. For protein-based therapeutics, you need to have shown target engagement in vivo. So we talked earlier about small molecules. Animal model data is not critical. For protein-based therapeutics, you do need to have that. And then again, IP is in place or obtainable. For stage 2, one or more potentially humanized protein-based therapeutics molecules should have been profiled so that you really have defined these further optimization needs. You need to have this strategy to reduce immunogenicity in place. You need to have shown therapeutic efficacy in vivo. So the last stage 1 was really about target engagement. Here you really need to show that it works in an animal model. Small-scale manufacturing needs to be available, and you need to start thinking about how I do this in a GMP facility. So manufacturing and scale-up needs to be feasible, at least from an intellectual perspective. And then you also need to make sure enabling technologies for either delivery or manufacturing are available and not blocked by exclusive IP. So if there's a company that has the greatest delivery system for peptides to the lungs, for example, but they have IP around this that you would need to take an exorbitant license from them or whatnot, that's going to complicate things, right? So you need to make sure that IP that you might use to deliver your protein-based therapeutic to the target tissue, should that be needed for your construct, it needs to be available to you for you to be able to license for reasonable terms. And then stage 3, you need to have selected a clinical candidate, again, characterized ADME properties, you need to have shown efficacy in an in vivo model. The clinical candidate has characterized toxicology properties, definitely need to have that GMP scale-up and characterization plan in place and have defined as non-clinical formulation. So then finally, in vivo gene therapies. Last year was the first year we allowed in vivo gene therapies to be eligible. We only had stage 1 at the time, and this year we've added stage 2 and stage 3 to this modality as well. So again, you need to have this target product profile. For gene therapy, the vector that you're using needs to only target somatic cells and avoid germline, just to avoid downstream complications. And you really need to pick a delivery platform that has a clinical track record. So we're not looking to fund your next best AAV capsid or whatnot. That's kind of out of scope. We're looking at projects that use currently available either viral or non-viral delivery systems, should you need a system. You need to have the target tissue and delivery route very well defined and need to have shown, again, in vivo efficacy. And then for vector-based therapeutics, the promoter and enhancer construct really needs to result in tissue-specific appropriate expression. And you need to have assays in hand to determine capsid quantification if you use a viral system. For oligos, you need to have shown structure-activity relationship. Again, assays should be available and validated. Toxicity and immunogenicity potential should have been assessed and again, have this de-risking strategy identified. And a small-scale manufacturing needs to be available. Likely this is the case because otherwise you wouldn't have done experiments with it already. But you also, again, need to start thinking about what does GMP manufacturing look like and scale up. And IP, again, should be in place or obtainable. And enabling technologies for either delivery or manufacturing are available and not blocked by exclusive IP. For stage 2, small-scale production of preclinical grade materials should be complete and available and addressing some scale-up hurdles. You need to have evaluated the biodistribution in a relevant animal model. The preliminary maximum feasible dose and dose-response relationship needs to have been established. And expression should have been confirmed via fluorescence, qPCR, immunohistochemistry, whatever is appropriate for your transgene. Integration or insertion site analysis has to at least been initiated to assess the profile and the risk. And really, you should have demonstrated functional rescue of a phenotype. Now, the preclinical safety program that's planned needs to include assessments for tox, immunogenicity, et cetera, and it needs to be in place. And there really needs to be a clear clinical plan now with potential biomarkers and endpoints identified. So again, talk to your clinicians, talk to your patients. What might a clinical program look like? And then finally, stage 3. For vector-based therapies, you really need to have shown scale-up and batch consistency across at minimum 3 lots. Again, manufacturing gets a little tricky with gene therapy projects, so that's why we're kind of harping on this. You need to have shown stability studies to show that the vector has adequate shelf life for clinical use. Minimal immune activation should be present at the clinical dose. The no observed adverse effect level or NOAL needs to be established with no systemic tox demonstrated in the appropriate species. Species differences start to play a big role again in gene therapy, so this is quite important. And then also critical quality attributes, so CQAs, critical process parameters, CPPs, release specifications for GMP manufacturing all need to have been defined. So this is really getting to the manufacturing bottleneck that a lot of these therapies have. Now, this is a lot of detail. We have a guidance document that I referred to earlier. We have all these eligibility criteria spelled out in those documents, maybe a little bit more detail. It's on these slides even, so you can look where does my project fit. I will also say that if you apply for, let's say, a stage 2 project with your preferred modality, we review it and we say, like, actually, you're better in a stage 1. We will not disqualify your project. We will just come back to you like, hey, we like it, but when you go to your full proposal, we want you to apply for a stage 1 approach. So don't be too stressed about picking your right stage, but it is important that you make your best fit as you can kind of guess, of course. So then here a little bit about the mechanics of the project selection process. So currently our call for pre-proposals is open. So we have a Salesforce-based grants portal. The deadline is Monday, March 16th. So for this pre-proposal, we're looking for high-level descriptions, non-confidential information only about the project, like the biological target, what's the project goal. And we have a programmatic review board. It's an internal group within TRxA and CPATH, and of course, the PKD Foundation to determine which projects would advance in the process. Then, if you get selected, we ask for a confidentiality agreement with your institution. So we will send out those requests in the middle of May. The deadline is about a month later. So getting, for us, a lot of these CDAs in place is quite a logistical lift. So we really hope your institutions will work with us as flexibly as possible. We've always managed to do this in the last couple of years, but it does add about a month to the timeline, unfortunately. But we want to make sure that when you submit a full proposal to us, that you're as confident as you can be that we will keep that data as, confidential as possible. So you make a secure repository available. At this point in time, you upload confidential information like your chemical structure, if you use a small molecule, your data package, and we will review that just to check for any IP red flags at this point in time. Most projects go through this step with no issues. And then ultimately, we invite you for a full proposal. The invitations will be sent out July 1. The deadline is about 6 weeks later, the middle of August. And throughout all of this, the full proposal process at least, the coaching and the mentoring is available from the TRxA team to put together a plan. Of course, we're not going to write the project for you, but we're happy to look at a draft. If you're struggling with how to position this, do we do this essay versus that one, we can kind of give you some feedback on that because we don't review the proposals. We have a standing scientific advisory committee. The membership roster is actually on our website. They will score the proposals, and then those scores get forwarded to, again, a programmatic review board and a PKD Foundation, who together will make the final selection of funding. So to further drive home the message that we're not just a money writer, check writer for you, what's really critical for our success is that we have monthly project team meetings, and we put together bespoke team member composition. So we ask people from TRxA, of course, being the largest CPATH organization, but then also, let's say it's a PKD project, of course, we will involve Chris or somebody he designates for scientific and disease experts. And if there's co-funding from other organizations, they will be part of those project meetings as well. Then we get feedback and advice from a scientific advisory committee. So of course, upon project selection, you get feedback on your proposal, but then also halfway through your project, our projects are for 1 year, so about the 6-month mark, we get the SAC to kind of look at the progress, give you some pointers, some adjustments if need be. Maybe they know somebody in companies that they may want to make an introduction to, and the same thing happens at the end of the project. And then what's also been proven really helpful is that we can provide connections to needed external services or expertise. So let's say you need to use a CRO to do some selectivity assays. We know kind of who to ask. We know what their rough cost is, and we can kind of help you direct towards those CROs that we have good experiences with. We don't manage the CRO money for you. We expect you to do that yourself. But again, we have introductions we can make. We know the people that respond to emails, right, that are easy to work with, and the quality of the work is appropriate. So we have time for a couple of FAQs, and then we'll open up the webinar for questions. Use the Q&A box, so not the chat box, but a Q&A box to ask your question. And Michelle is monitoring those questions. At the end of this FAQ section, we have some time to answer questions live. But let's dive in. So who's eligible for TRxA funding? So really faculty at universities and nonprofit institutions anywhere around the world. We do ask for faculty members or the equivalent to be applicants and not necessarily trainees, just because of stability of employment. Trainees, you're probably going to go to a different institutions after your project ends. So that gives us some uncertainty that we want to eliminate. So you need to be faculty in order to be eligible. And then there's always questions like, well, what are the institutional commitments that come into play when we get funded? So we, of course, expect to engage in a grant agreement. So this is a very reasonable project agreement that we will put together. We actually have a template of this agreement available on our website. In your grants and contracts office or your technology transfer office, I'm sure they will be really happy to look at this template. We don't pre-negotiate agreements, but they can kind of look at, are there any red flags? Thus far, we've been able to negotiate agreements with institutions all over the world. We're very reasonable and flexible, but still, it's important that you know that what are the kind of the strings attached, so to speak. So wanted to reiterate what therapeutic areas are eligible for this year's awards. So rare and orphan diseases, brain health, pediatrics, and of course, polycystic kidney disease on a dedicated PKD Foundation track. And then we have sometimes questions around what qualifies for the in vivo gene therapy. So I gave you some examples. Again, to reiterate, you really need to use a delivery platform with a clinical track record. And some things that are excluded are exosomes as delivery vehicles, and there's a lot of variation in there, and still some questions around how do you define critical process parameters around those and so on. So for now, they're out of scope. We also do not fund replication-competent viral vectors like adenoviruses, VSV, HSV, etc. And then, of course, you cannot target the germline cells. Indirect costs or overhead are allowed, but it's limited to 10%. So, and also pass-through costs to service providers or like CROs or consultants, that's not eligible for indirect cost, but we do allow 10% on the rest. So then the question is sometimes asked, like, well, do I get all the money? So yes and no. So we award the funds to the project. So this may not necessarily all go to your lab, maybe part or maybe even most of it may go to, for example, CROs or consultants. So we do allow the use of CROs and consultants. We actually encourage you to use this really specialized organizations that can really help accelerate your project. So that is completely allowed and encouraged. And then whatever you and your lab needs to do to make the project a success, of course, you can ask for funding for that. So allowable costs include costs associated with defined project tasks like lab supplies, animal procurement, salaries, core services. What's not allowed are general lab expenses, travel, equipment, tuition, or IP prosecution and maintenance costs. The latter is because we don't own the IP. It's going to be owned by the university. So we expect your university to carry those costs. And then sometimes we get the question, do we need to worry about having other funding sources from our project? And now, of course, you can't double dip, right? You can't have two funding sources for the same project, but you can have actually money for different aspects of a project. And we actually encourage it. So we really like to partner with other funders to accelerate your program. And again, we have, of course, the PKD Foundation dedicated track this year, but then also examples of those co-funding partners for this year with Project CASC and Cerebrum DAO. And then once we see all the projects that come in, we may reach out to even other additional funders about, hey, this is, let's say, a prostate cancer project. Do you guys fund prostate cancer? Are you interested in, well, A, taking a look at it from a diligent perspective, but then B, maybe you want to together support this program so that we can do things better, faster, and more efficient. And then we talked briefly already, how do you find the right CRO if you need one? So really TRxA can help with that. We know many CROs all over the world. We don't have geographic restrictions necessarily. Your university may, so check with them. And of course, the management of the contract with the CRO is through your institution, but we can help manage the work to be done. Sometimes we get asked questions if you need quotes for your pre-proposal. You do not, especially not for early stage work. And then we can really provide some ballpark numbers as well for standard drug discovery assays like selectivity panels, PK studies to help you build your budget. For later stage work, let's say you really need to have a CRO do scale up and impurity profiling for your compound, then you probably need to have a quote because that amount of money can vary widely depending on which CRO you use. And it also, you need to know how much that kind of work costs in order to appropriately position your project. And then we briefly touched on this already in the beginning, whether you need to have IP filed on any compound before you apply. There is not such a requirement, but we really want to make sure that the composition of matter is eligible for protection. Now, we do expect any IP to be filed, either has been filed or will be filed, that is filed internationally. So not just the US. From a business perspective, that it tends to have a lot of limitations. It will be really hard to get funding down the line if you only have protection in the US. So at minimum, we're looking for US and Europe, and ideally several countries in Asia as well. So then some details, how do I apply? Well, we have the Salesforce portal. So create an account in this grant portal, and the link can be found on the website. Our website is c-path.org, and programs and TRxA. If you Google it, I'm sure it will pop up as well. Now, what's important is that this portal will automatically close at midnight in your local time zone or wherever your computer thinks you are. So it's been every year things are going smoother and smoother, but sometimes there are some account issues that you're like, hey, I can't get logged in. I need to reset my password. We can, of course, help you with that. But please start early so that we can mitigate any of those technical submission issues. And then your chance of success, I'm sure you're curious about that too. So we anticipate funding between 3 and 5 awards this funding cycle. There's really not a predetermined number of awards in certain areas of therapeutic interest, modality, or funding level. So it really depends on the quality of the science that we get in. So the chance of success, so it differs from year to year, of course. We can't, project the future. But for 2025, some data, we received a total of 79 pre-proposals. Of those, 20 were selected to submit a full proposal, so about 25%. And of those, 25 were funded, so 25%. So that gives you hopefully a bit of a measuring stick to know what your effort level needs to be for this particular process. I think for us, having to split up this pre-proposal and full proposal stage is really helpful because the pre-proposal shouldn't take you a whole lot of time to put together. It's not going to be weeks and weeks of painstaking work. All of our proposals are question and answer based. So we ask you a question, what is your therapeutic indication? And then you have 200 words to answer. Right? So it's very tactile, very clear what you need to put in several places of the application. We also have some example applications on our website. So take a look at all those materials to kind of see what's needed for each stage. And then if you need advice, if you have questions, we're available via email at TRxA@cpath.org. We do ask that you ask your question via email first, just from a bandwidth perspective. When we get lots of interest, we would love to hop on a call with everybody who asks, but just logistically, it becomes very, very challenging. So we'll try to answer your question via email. If we cannot get this resolved easily, then of course we will get on a call and help you with your application. But just know that we are monitoring this email box every day, very actively. Kyla is looking at that, and we really stay on top of things. So then here's the last slide. Next steps, create an account in the Salesforce grant portal. Again, you can find the link on our website, and then submit your proposal by 16th of March before midnight, your local time, or your computer's local time if you're on a VPN. So really start early. It really helps to not be stressed about technical aspects of things, and we're really looking forward to see what comes in. So I'll save this. I'll have this slide up for a few more minutes while I invite the rest of the team to come back on camera, and we can start looking at some questions.
And we have lots of questions, Maaike.
Of course.
So we'll dig right in. The first question is of 2026 RFP. How does Michelle, you were breaking up a little bit for me. Can you maybe repeat the question? Oh, you're totally frozen now. Maybe, Mark, I see you moving. If you can take a quick... Oh, Michelle's back. Here we go. Can you repeat your question, Michelle? We're ready for another question. Okay.
No, sorry, we missed your first question because you froze up.
I froze up. So for the purpose of this RFP, how does TRxA define rare and orphan diseases?
Let's see. I'll start with that one. There's different definitions all over the world. I would say look at the either FDA or the EMA definitions, and either one would be fine. If you are ultra rare, let's say N of 1, that's going to be a little bit more difficult to kind of build a program around. But if there's at least a few hundred patients in the world, then we would definitely take a look at that, up to, let's say, 10,000 or so.
Great. Okay, we have a...
Oh, Michelle's freezing again. So, Mark, if I can ask you to maybe jump in here and look at the questions and monitor those a little bit.
Yeah, I don't want to answer this one, Maaike, so I'm going to throw it to you. So are delivery platforms for CRISPR and prime editing using RMPs, i.e. not DNA vector-based approaches, eligible?
But so the CRISPR platforms that are not what based?
Using RMPs.
RMPs. I don't know what RMPs are. So
no, I actually gave my email, so this might be a...
There you go. Yeah. So if we don't know the question, please
I
email us and we'll figure it out.
Okay, I threw it to you.
Okay, am I back online?
Yes.
Okay. So can you elaborate on what would be in scope for brain health proposals?
Sure. So we're looking at neurodegeneration in particular. That's definitely in scope, but also psychiatric disorders. So really anything that has to do with the brain that would be considered eligible. But I would say our sweet spot is probably neurodegeneration. But again, don't exclude yourself to that.
And feel free to reach out via email if
Of
you'd like
course.
some clarity before you put in the work.
Yes.
Okay, and so I have a question at the PKD Foundation. Chris, would the PKD track cover all types of PKD?
Yes, so the short answer is that there's no restriction for AD-PKD specifically. Certainly, if you have an AR-PKD proposal, that certainly would be entertained. I think we're going to be evaluating partially based on like impact of the proposal as well as feasibility. So there certainly are differences between AD-PKD and AR-PKD, and there are certainly potential therapeutics that cross both. And it's also worth mentioning, since I didn't mention it earlier, that there is no approved therapy for AR-PKD currently. So there's certainly an appetite for that.
Great. Thank you. Mark, do you have another question for us?
Yeah, is past applicants, are they eligible to reapply?
Absolutely, yes. So we have actually 2 success stories of people that reapplied last year and were able to get funded in this most recent funding cycle. And that is also so A, keep at it. Don't lose hope and keep applying. But also we try to give actionable feedback to you, both at the pre-proposal stage and the full proposal. The full proposal definitely you get full reviewer comments. For the pre-proposal, it's a little bit again based on volume. We try to give you as much feedback as we can as possible, but we don't guarantee that we will have paragraphs for you. But we will at least give you some reason to make sure that is it worth your time to resubmit, or is the project kind of a no-go for whatever reason? It just doesn't fit within our parameters. We will give you some guidance there so that you know whether reapplication is a good use of your time.
I've got another one for you, Maaike, I think. So what are your thoughts if a project is early stage and has been through a phenotypic screen and some optimization, but target ID still needs to be completed?
Ooh, that's a good one.
Yeah.
So very good question. So we actually have funded a project in the past where a phenotypic screen was used to identify chemical matter. The PI was in the process of identifying the target. So I think it depends a little bit on how far along you are in that target identification process. And if you have an inkling, then I would say that's probably going to be our scientific advisors are probably going to be comfortable enough to look at your program. If you really have no idea yet, then I think you need to make a very compelling case that the assay cascade that you've set up is going to be sufficient to tease out project compounds that are going to do the thing that you wanted to do. But ultimately, from a commercial and business perspective, there is still some bias out there that target-based programs are considered less risky. So we need to be cognizant of that reality, whether it is, fair or not. It is the world in which we live. So I think the bar for the quality of the project when you've used a phenotypic screening and don't know your target is going to be a little bit higher than if you have a target-based project.
Okay.
It looks like Michelle's back. Welcome, and you're on mute.
Maybe I'll there's another question. How do we define success for a funded project? I.e., what's the valuable inflection point? Maybe I'll give Maaike a rest. So really, I think applicants need to think about the change that's going to happen during the funding. So really, what can we help drive in terms of change that wouldn't have been there if you didn't get that funding? So for early stage projects, maybe you're not going to get to IND. Maybe for stage 3, the IND is a good inflection point. But really, things like proof of concept in an animal, something that's going to encourage other funders to maybe take that project to a point where other funders are going to become interested, where we can generate that data, I think is a good inflection point.
Yeah, I would.
Sorry, and then I'm going to have another question for you, Maaike. It's a very broad question, but what makes an application competitive?
The magic sauce.
Yeah.
So I would say, based on a couple of years of doing this now, the projects that tend to do well already have an interdisciplinary team together in whatever form. So some may be informal, some are formal, where different co-PIs are part of the project team. But especially in the early stage programs, having somebody with biology expertise working with somebody who understands the clinical problem, so you're having a clinician on the team. You are working with somebody who understands the modality, so it could be a medicinal chemist if you're working on a chemistry small molecule project. It could be somebody who really understands protein engineering for protein-based therapeutics and so on. Those kind of groups tend to already speak this interdisciplinary language because they've worked together with each other for a while and know that, kind of what the terminologies are. That tends to come through in the application. So there's no requirement that you have an interdisciplinary team like that in place. But I will say, if you're not currently in one, I would highly recommend reaching out to your colleagues, either within your institution, outside. We have projects with people from a couple different universities. That's totally fine as well. But talk to some people to give you some ideas and advice on what the project progression pathway should look like, and ultimately what your target product profile needs to look like as well. And it kind of shows in the application when people have done that exercise. The other aspect of that is if you've engaged with your technology transfer office. Again, no requirement there, although at some point in the application process, we do ask for a letter of support from your tech transfer officer. That's at the full proposal stage. The reason for that is, if you've engaged with your tech transfer officer, they have probably asked you questions around, why is this new? Why is this novel? How do you compare with standard of care? Why does anybody want to invest potentially money into this project to make sure that new therapies become available to patients? What's sometimes a bit of a pitfall of academic investigators is there may be biology or pathway driven, and you may be a 20-year expert on, a certain kinase pathway or whatnot, and you try to find a disease that fits into one of our criteria so that you become eligible, so that you can play with your pathway more. Well, sometimes that's fine, and it's actually a really good solution for a problem that this disease might have. But sometimes it's more a solution looking for a problem rather than a solution to an existing problem. So conversations with your technology transfer office will probably have clarified that, and you may have some language around why your project is cool, unique, and investable. And again, that comes through in the application when you write it up, because we're going to ask you some questions around that. So again, talk to a lot of people outside of your immediate silo, and that will really improve the quality and the success of your application.
If we've got time for one more question, it's about the IP and the spin-off. So the boundary between a spin-off project and an academic project is very thin, which is very fair. But for what reasons did you decide to fund only the academic projects? Maybe this is, you don't have to give a history, but we've got a lot of questions on the spin-off question.
Yeah, and we're kind of somewhat sad that we have to put that hard line in the sand. This was probably our first year where we had an annual RFP where we still were kind of unsure. And in talking through where we had an occasion where the IP was already optioned to a spin-out company. And we had a lot of internal conversation within CPATH as a whole that we ended up deciding not to allow that. I alluded to it a little bit earlier, but just to reiterate some of that. Critical Path Institute is a neutral convener to pull together industry, academia, patient groups, NIH, academics, etc., into these places where we're really in a pre-competitive space. For us, that is the North Star as an organization as a whole, to be in this pre-competitive space. If we, as TRxA, as a funding program, were to fund one company, even if it's a startup, there could be a perception that we are now abandoning our neutral stance as an organization. And because that North Star is so critical for us, that neutrality, we decided to, again, put that hard line in the sand and not go that route. Again, we understand sometimes it's almost just startup in name only because you filed the paperwork so that you can apply for STTR grants or SBIR grants in the US. But unfortunately, it's the reality in which we live that we need to deal with. And that's why we made the decision to protect the organization as a whole and its reputation as this neutral convener that doesn't have a stake in the game. And that's why people trust us with their data, with their opinions and their scientific input. And that's why it's so important for us to limit it to academics and nonprofits. Okay, maybe one last question that I know people would have wondered about. Kyla, can you talk about whether slides are available and the recording will be available?
Yes, absolutely. Since you registered for this meeting, you'll be receiving a copy of the recording in your inbox, and it will also be available on our website. The slides available for download as well, and we highly encourage you to share it with anyone that you think would be a good fit for our program.
Fantastic. All right, well, that concludes our webinar. Thank you all for being here. We really welcome to see your applications. Again, if you have questions, please reach out to us. If it's a PKD specific question, we of course are in constant communication with Chris. And Chris, thank you for being here and being part of this initiative. I'm really excited about it.
Yes, I'm excited as well. Thanks so much.
Have a great day, everybody. Thank you.